So, good morning, everyone. So, I welcome you to the final session of this Data Science for the Biologist program. So, in this final session, we will talk about how we can design a research project using available NGS data and employing T-Bioinfo platform. So before going into today's session, let's review what we have covered during this program. So we started with the history of the genomics data, that how currently a discovery of the DNA structure led to the thousands of the sequencing of the thousands of the genomes. Then we learned about important characteristics of a data that makes a data into a big data. Those characteristics include its volume, variety, value, veracity, and velocity. Subsequently, we have learned about various aspects of a data-driven approach and their importance. Next, we learned about different omics layers of NGS data. Then we understand about NGS sequencing approach. Next, we learned about how the NGS data can be stored into the different file formats, that is FASTA, FASTQ, GTF, SAM, and the WAM files. Then we get an overview of DNA and the central dogma of the molecular biology. Next, we learned about different NGS sequencing techniques. Subsequently, we understand about different types of sequencing reads based on its length. It could be a small reads, it could be a large reads. Then we learned about type of mutations that include point mutations, which are further divided into substitution, insertion, and the deletion. Then we looked over the mutation that were generated via the rearrangement, like inversion, deletion, etc. Next, we learned about type of mutation based on the impact that they have on the genome. Then we learned how a change, even at a single nucleotide label, can be good enough to change the type of protein that it expressed by changing the coding sequences or the codons. Next, we understood how a mutation in the key genes can promote the tumor formation. Then we demonstrated to you how we can visualize the sequence alignment with the reference genome using T-Bioinfo platform. We have also learned how we can perform various mutational analysis. In the next session, we go through the overview of metagenomics and metagenomics techniques. Then we also learned about 16S RNA sequencing technique and its importance, how it can be employed for the taxonomic classification. Subsequently, we demonstrated to you how we can perform metagenomics data analysis using a simple pipeline that is available at T-Bioinfo platform. Next, we demonstrated to you how we can perform and analyze 16S amplicon data employing data to pipeline at T-Bioinfo platform. Then we learned about what are alpha and beta diversity, and how we can analyze metagenomic samples and what based on the alpha and beta diversity what they represent. Next, we get an overview of RNA transcription process and what are important steps. Then we also understand what are different techniques that are employed to measure mRNA abundance that include RT-PCR, northern blot, microarray. Then we get an overview of RNA-seq technology, how we can quantify and using the RNA-seq. Then we get an overview what are important steps that are required for RNA-seq data analysis, including pre-processing, mapping, and quantification of RNA expression. Here we also learned about what are different algorithms that are required to perform a particular step for RNA-seq analysis. Next, we get in next we demonstrated to you how we can generate a gene expression table from the raw sequencing read using a simple demo RNA-seq pipeline at Bioinformatics platform. Then we understand about various aspects of a gene expression table, what it represents, what are the row, what are the column. Then we also demonstrated to you how we can visualize our gene expression data in the Excel. Once we have a gene expression table, so what are different statistical methods are available? So we go to the t-test. Then we also demonstrated to you how we can perform t-test in the Excel. Then we demonstrated to you how we can perform differential gene expression analysis for the raw read code data using DSEC pipeline. So next we go, if we have more than 2 factors to consider at a time, so how we can perform factor regression analysis. Next we learned about once we have a gene expression table, so what a box plot represent and how we can measure various statistics about of data using simple box plot. Then we also learned about how we can perform log scale transformation on data and what's its significance. Then we also learned about how the log scale transformation impact the data and what is the importance of normalization. Then we go to the another normalization method and understand its importance, that is quantile normalization. And we have also demonstrated to you how the normalization of our data affect the biological meaning in the data. Then we get an overview of machine learning techniques. Then we go to the unsupervised machine learning methods, out of which we first focus on principal component analysis, and we understand what are how we can visualize patterns in our data using principal component analysis. Then we demonstrated to you how we can perform principal component analysis on a Bioinfo platform. And eventually, we have also demonstrated to you that how we can visualize pattern within a data based on the scatter plot that is drawn using principal component first and principal component second. Next, we focused on the another unsupervised machine learning method, that is hierarchical clustering, and we demonstrated to you how we can perform hierarchical clustering and what they represent. Then we get an overview of supervised machine learning technique, and we learned about what is the training and the test data, and what is the difference between these two types of data. So then we also understand out what are different supervised machine learning algorithms, and what are its major category like classification and regression. Then we also provided you an example that like random forest decision trees, SVM, etc. Then we also get an overview of binary decision tree. And in the last session, we also go through this random forest methods. So today we will first we will demonstrate to you how we can execute random forest on T-Bioinfo platform and what it represents. And during this whole program, we have tried to provided a demonstration in the R, that how you can visualize your data and how you can perform dimensionality reduction. So let me see how much you understand during this program, and let me ask a few questions from you. So which changes sequencing technique have highest coverage? A, targeted; B, whole genome; C, whole exome. So during genomics, we understand different NGS sequencing techniques. So which NGS sequencing technique have highest coverage? Yes, Bharat, absolutely correct. It's targeted. As targeted sequencing is focused on a particular very small region. So usually it has the highest coverage. So which type of mutation inserts stop codon in the sequence? So which type of mutation insert stop codon in the sequence? Yes, Atma, you are absolutely correct. It's a nonsense mutation. So the next is, it is a statement. You just need to tell whether it's true or the false. Frame shift mutation are always the result of mutation only at a single nucleotide position in the sequence. True or false? Yes, you are absolutely correct. So it is false. So which of following parameters provide us information whether mutation is significant or not? So this is based on the results of mutational analysis that we run at T-Bioinfo platform. So the A, position of mutated nucleotide at a reference genome; B, mutated nucleotide base; C, somatic score; D, none of above. So Bharat, yes, you are absolutely correct. It's somatic score. Which of following are the two algorithms that can both be used before mapping? A. PCR clean and trigonometric. B. Clean primer and isolation. C. Eco and SR. D. Butai and PCR clean. Yeah, it is PCR clean and trigonometric. So PCR clean removes the PCR duplicates and trigonometric removes the adapters. What types of the files are used and the input for the RNA-seq pipeline? A. FASTA or the FASTQ, which include DNA sequences. RSCM or TXT file, which include DNA sequences. C. SVL or URL file, which include RNA sequences. Excel files, which include RNA sequences. So, what type of files are used as input for the RNA-seq? Yes, Atma, you are correct. So, it is the FAST and the FASTQ file, which actually include DNA. It could be because it derived from the RNA. So, usually we call it cDNA, that is complementary DNA. So the next question, in general, researchers don't use t-test in RNA-seq research. Which of following is the reason for that? T-test can be applied on a data having normal distribution. B. T-test can be applied on RNA-seq data that have read count values. T-tests are not suitable for hypothesis-free research. That is a common approach in a whole transcriptome RNA-seq. And D. T-test can only be used for categorical data. So the t-test can be applied only on a data that having a normal distribution. So the next question, you have run a t-test on 4 genes in Excel using this particular formula. So in the column W, to see if their expression is significantly different in different breast cancer subtype, which of following genes have significant different expression? A gene involved in cell proliferation with a p-value 0.04 in the column W. B, a gene involved in metastasis with the p-value 4.0. C, a gene involved in the tumor promotion with the p-value 0.44. And D, a gene involved in menstruation regulation with the p-value 0.04. So which one is correct? So I'm just giving a hint that t-test is as significant p-value should be considered. So Bharat and Atma, you both are incorrect. P-value, how much p-value considered as significant p-value? So yes, Utkarsha, you are absolutely correct. So option is D, that is gene involved in menstruation regulation with the p-value 0.04. So as in t-test, p-value less than 0.05 is considered as significant. So the option D is correct. So the next question, which of following provides summary statistics of the data? A, scatter plot, B, bar plot, C, box plot, and D, histogram. So yes, Bharat and Atma, you are absolutely correct. It's a box plot from which we get various summary statistics like first quartile, third quartile, median value. Then we also get the information regarding the outliers. So the dendrogram is the output of the following method. A, k-mean clustering, B, t-test, C, principal component analysis, D, hierarchical clustering. So Atma and Utkarsha, you are absolutely correct. It's a D, hierarchical string output, but we get a dendrogram. So the next question, which of following is the supervised machine learning method? Option A, PCA. Option B, t-test. C, hierarchical clustering. D, random forest. So which of following is the supervised machine learning algorithm? PCA, A, B, t-test, C, hierarchical clustering, and D, random forest. So yes, you are absolutely correct. It is random forest. And today first we will demonstrate to you exactly what would be the output of a random forest method. So before going into how we can design a research project based on NGS data and what should be the approach, and let's just cover the random forest that we were talking about in the last session. We were talking about different supervised algorithms, and one of which is random forest. So let's go to the demonstration how we can run random forest on T1 platform. So meanwhile, let me share just two files. So if you remember, for the supervised machine learning, we must have two type of data. One is training, the other one is test. So in the training, we have a labels or the class, and while test, that lacks those labels. So just let me share files with you. So I'm just pasting the link. This is test file. Then we have our training file. So just download these files on your system. Once you're done, just let me know. Meanwhile, let me just show you exactly what these files contain. So this is our training file. If you look at here, we have IDs of the samples, then we have the class label. So here we are using a cell line data. So breast cancer cell line data, where we have different breast cancer cell line subtypes like luminal. If you look at here, luminal, claudin low, then we have normal like and basal. So these are the classes of the samples, and then we have a gene expression table. If you look at the test data, here we have only ID of these samples and it lacks the labels of this test data. So once the supervised machine learning learn the class labels in the training data based on these features, then it will predict the labels of the test data. So now let's go over T-band for platform. So once you are there, just let me know that. Once you're able to log in, able to download these files and able to log into the T-band for platform. So apart from Atma, who else is able to log into the server so that we can proceed and demonstrate to you how we can run random forest or any supervised algorithm on T-band for platform. So here, if you go to the data mining section, okay, so no issues. So here we have, it's okay, Bharat. So under the data mining option, here is the first module, that is supervised analysis. So if you click on this, then we will direct it to this interface, and where first option that we are getting, that is file upload. Here we have to provide the files. So when we have to upload these files, before uploading a file, let's make sure here there should not be any blank line. So just let me check the file. So I have already shared the link with you. So let me just save this file. So just upload these files. So then just go to the group selection. So here we have to drag our files into training and the test group. Then we can to continue. Next is start. So here the different algorithms already highlighted. So you can choose any of these algorithms. So for the moment, I'm choosing random forest. So the random forest, if you remember it, it requires first it generate number of trees. So then the out voting of the each of the tree is then voting from each of these trees actually decide the label of or the class of each of the samples. So finally the maximum voting, if we maximum trees are majority of the trees are saying that the label of class is particular label, so then the class label is decided based on that majority voting. So here you can decide the number of trees option. By default is 500. So to run any pipeline, if there are large number of trees, although they will provide you a better performance, but the again the issue might could be that like that if there is large number of tree, it would be computationally expensive. So here I'm just uh, reducing these trees to the 100 so that it won't take much time. So then we are ending this pipeline. So it will take a 2 or 3 minutes to complete this pipeline. So the random. So it will take 1 or 2 minutes. So whether Atma, are you able to complete your pipeline? Then Utkarsha, what about you? Okay, so is it some issue? Okay, okay. Okay, that's fine. So it will take a 1 or 2 minutes. So meanwhile, just let me tell you, from the random forest, what we are going to get one first, of course, the prediction labels for the training data as well as the test data, and we will can compute the performance that is in terms of an accuracy. Then we will get as also output in terms of that is feature ranking. So the advantage of random forest and stepwise LDA that apart from performing or develop a prediction models, they also rank the genes. So as progress, we can see that progress 33%. It will take a 1 or 2 more minutes. Yeah, it's already completed. So let me just show you the output, what we have obtained. So here the first is confusion matrix. Let's see what it represents. So let me just paste it in the Excel. So here what we are getting, like these are the true labels and these were the predicted labels. So for example, here we have a basal and basal sample which is also predicted as basal. So we can highlight it. Then the next is, this is Claudine samples. So 4 Claudine samples are actually predicted as basal samples. So none of the Claudine sample is correctly predicted. Then if you look at the luminal, so there are the 20 luminal samples which were predicted as luminal. Then we have normal samples, and here out of the total 3 normal sample, out of which only 1 normal sample is correctly predicted. So in the diagonal you will get the exactly how many how many samples are correctly predicted. So this is indicated the class error. Here, as we know that all the 11 samples are correctly predicted, so the class error is 0%, but the accuracy for the basal sample it's 100%. Then for the Claudine Liv sample, none of the sample is correctly predicted, so it indicated the class error is completely 1 or 100%. Then luminal samples are correctly predicted 20 out of 20. Then we have normal like samples, out of the 3, 1 sample is correctly predicted. So the overall accuracy of this model on the training data, what would be that we have to plus this total. So like 11 plus 20, that is 31 and 32. So the 32 samples are correctly predicted out of total 37 samples. So now you can compute how much accuracy it has. 31 out of the 37 samples. So how much accuracy it would be? So it is, you have to just compute a simple percentage. So can you tell me the accuracy of this model? So 32 out of 31 out of total 37 samples are correctly predicted. So the accuracy that we will obtain, that is nearly 86%. So this is the first output. If you go to the other output, next is feature importance. So before the going to the feature importance, let's the result for the test set. So here, if you download this file, so here it will predict the labels of each of these samples. So the class of each of these samples. So this is the predicted class. So then we have this output, that is feature importance. So how you can select the features? So it rank the features. So let me just copy it in Excel. So here you have this column, that is mean decrease accuracy. So you can select only those genes which affect the accuracy of a sample, accuracy of the model. So what we can do, how we can select the features, set a this filter, and then we have to just largest to smallest. So you must consider only those features which affect the accuracy of the model. So you can now make a cutoff, like if you want to take a more you want to take only those genes that have actually affect at least 1% accuracy, right? Similarly, you can take a positive and the negative value, and you can ignore other genes. So you can then you can develop prediction model based on only the selected genes. So this is the it provides you the importance of each of these features. So how they affect the performance. So you can rank these genes and try to identify only important genes instead of considering all the 10,000, 20,000 genes. So you can consider only the top genes. So this is what the about the random forest model. So now let's go over the presentation again and let me show you what are important steps when you are designing a research project. So what should be the approach when you're designing a research project, bioinformatics research projects? So the any research projects, it start with the when you first find a frame a problem. Once you identify your research question or problem, then you go through the literature review to understand its background and how you can design your and address your research query. So once you understand how you are going to address, then next step is collect the raw data that is needed to address your research question. Then you explore your data and pre-process it for any analysis. Then you perform in-depth analysis to derive a biological significance from the data. Finally, you summarize your results in the form of a research report or a research publication. So how you design your research query? So once you are going through the journal background overview, then you try to find out what are different available sources, and when you are going through the review of literature, and you try to find out what are important loopholes or gaps that needs to be addressed. So then you also understand what are important questions that are associated with your research query, and try to find out what are already important controversy regarding a particular research question, and then you define your specific hypothesis. So this is what the simple approach who you choose how you choose a topic for your research. So for that, first you are interested only the topics. Like for example, you are interested in a particular malignant condition. So you understand what you already know, topic you know and you are care about. So then to try to find out what are the important interesting audience that are actually going to understand your research query. Are there enough people that actually this particular research topic is important? That is whether, for example, you are choosing some problem that is actually not concerned with the any of people, like this is not a research issue for much people, so you cannot consider such a, usually you consider only such problem that actually affect a lot of people or lot of population. Then and you discuss this topic with your colleagues and with your other research people, and then you try to find out whether there is sufficient studies and the data is available. So then you try to go up public repositories and try to understand whether there is any data set is available for a particular study. So now let's understand what are different resources that are available for a big data. So whenever you design your research question or you have just plan or decide a research topic, So the next is where you can actually go for literature review. So one of the major repository for that is PubMed. So here, if you have any query, and we will also demonstrate to you, so how we can go through the review of literature and try to find out different issues. So to understand and go through the review of literature, PubMed is the best resource. Then what are different resources from where you can obtain a data? For example, if you are interested in a transcriptomics data, one of the major repository for that is available. For that is GEO, that is Gene Expression Omnibus. Then if you are interested to the raw sequencing read data, so then the one of the dedicated repository, that is Sequence Read Archive, so that can provide you the read sequencing data. Then there is another project, that is repository, that is Bio project. All these repositories from the NCBI. So then if you're interested in a particular topic, like if you're interested in a apart from this, then another repository, that is Array Express. Then if you're interested in a cancer related data, then a dedicated repository where there is data available for nearly 33 cancer types, that is GDC data portal or the TCGA. So the difference between GDC and TCGA, in GDC portal, apart from the TCGA data, there is some of another repository like Target. So you will find the data for that as well as for the Jenny. So then we have another repository, that is UCSC cancer methylation system. Then if you're interested in the mutational data for the cancer patients, then it is a Cosmic. And then there is if you're interested in the protein data, then there is Human Protein Atlas. So if you're interested in some viruses, then one of the major repositories, that is NCBI virus. So like if you're interested in Ebola or COVID-19, then the sequencing data, sequence data, that is a complete whole genome sequence in the format of FASTA files is available at NCBI virus. Now let's go over one of these repository and try to understand how we can proceed for a research problem. So the first, once you have a question, you already have a research topic. For example, we are interested in COVID-19. So what's the background that we have? So if you look at here, so you will find like nearly more than 85,000 publications are there. So then you can just go to this, and if you're interested in a particular problem, then you can also go to the advanced option. So for example, this but is this advanced option indicate? So for example, we are interested in SARS-CoV-2. So here, if you are interested, and like for example, you can select a particular option. Like title, we want to make a query where SARS-CoV-2 is present in the title and the abstract, and then you can add another query and make a complete query. Like for you, for example, you are interested in some drug treatment, that is so you will find like you can make your specific query. Like here you already get a SARS-CoV-2 drive JAK1 2 dependent local and systematic complement. So then you will find the publication related to that particular query. So you can make the different queries, and exactly you will find exactly the research question that you are interested. For example, you are just interested in the treat ruxolitinib with the treatments, so the drug treatment. Here you will get the query regarding that, like 261 article, like that. So then you can, if you are interested in a particular topic, so then you must go to the first, go through the review articles. So always try to find the recent review articles that will cover the maximum information. So here you will get the different review articles, and then try to go through all those review articles and try to find out what are still the loopholes that is associated with your particular research query. In the advantage of review is that you will also get the information of already published work, like already what has been done and what needs to be addressed. So the whenever we proceeding for any research problem, we must go through the review articles so that we understand what was the background and still what is the loophole. So always go through the recent review articles. Once you understand and make your hypothesis, then try to go through the recent research articles, and exactly how you can plan your research, how you can design that. So this is how you can search for the puppet. The next is how you can search your data sets at the different repositories. For example, we are at the Geo. So let me just go over there. So here, for example, you are interested in COVID-19 and try to, let's see how we can download data from the Geo. So if you look at here, first at the right side, here you will get this option that is organism. For example, you are interested only on the human sample. So like human, you must to obtain only those studies where there is human samples involved. So you can select that and filter out. So then if you are, the next is what kind of study type you are interested, what kind of the study that you are interested. So under the study type, from here under on the left side, here in the third option, that is study type. So you can go to, if you go to the customize, you can actually select and filter what kind of data that you are interested. For example, here is already given RT-PCR, SAYS, SNP array, high throughput sequencing. For example, I am interested in here to obtain a data set which contain expression data from the high throughput sequencing. And you can unselect others. So I'm unselecting methylation profiling. So then we can just go to the show. Here you need to click on this. Then here you will get the various data sets. So we can sort these data sets based on the number of the sample, like high to low. I have already added this filter that is sort by the number of the samples. So now if you are interested like in a different sample, so even if the first study, if you look at here, so you can directly, you will get it, if you go to this study, and you will get like here already 726 samples are available. So then we can go to the individual study. Then like here, the diagnostic host response biosignature for COVID-19 from RNA profiling of the nasal swab and the blood. So we know that this is the blood samples that have total 404 samples. So then whenever you go to a particular study, you first, you will find the details regarding summary, exactly what they have done in that study. And then you will also get this under the overall design, exactly how many samples are there and how they done that. And then you can also go through the particular publication that they have worked on for they have authors have submitted their data, but publication yet to come over. So here publication link is not available. Otherwise, under the citation, under the citation, you will get the even the publication associated with the particular research query. So then this is how you can search already your data from the whole. So then here under the supplementary file, usually they provide some already processed data, and then apart from the processed data, under the series, this download family, this is the series matrix file. So this series matrix file actually contain the data set that is already processed with the clinical characteristics of the samples. Then they have also provide the links to the other under the relation, like they already provide the link like the bioproject or the SRA. So this is how we can actually search a data sets from the public repository like Geo. So then in the Geo, if you are interested, already you know that there is a particular data. So then sometimes we go through a particular publication, and then they provide you something like GSC ID, that is Geo ID. For example, I have one data set. I have gone through this publication, one of these publication, and have already find out that they have done ruxolitinib treatment. So different they have this gene expression data once they treated the cell line samples with the different drugs including ruxolitinib. So as we know that ruxolitinib is one of very important JAK-STAT inhibitor, so that is currently employed to cure this COVID-19 patients. So let me just find the data set for that particular. I have that GSC ID, so I can query this GSC ID here, that is 175 147 507. So let me just copy it here as well. So you can directly under the geo session, you can find, you can query for if you already have a GSC ID. For example, I'm just copying this GSC ID with you. So I have just gone through this publication before this session, and I have gone through that. Here they have cell line samples. So from the human lung, primary human lung epithelium cells, and cell line samples that are actually treated with the different drugs. So then if you go through this publication, here they have already summarized what they have done, and they have actually used various chemokine related drugs, and then they have also provided what are different drugs and how many samples are there. So then they have also provided, as I have told you, as we can find the publication that is associated with this particular data set. So they under the citation they have provided that link. Then here under the supplementary files, usually they already provide their process data. So here the two types of samples are there, that they have human samples, and then they have Stella pastoris furo. So I'm only interested in the human sample. So you can directly download this read count values. So you can download data from here. So then they have also provided the links to the SRA. So if you are not interested in the already processed data and you want to process your own pipeline, what you can do, you can directly go to this SRA. Already they have provided a link for that. So if we are go to this SRA, here we will get that directly to this SRA. But before, how you can obtain this read values? So what you can do, you can go to this send to and create a file, and then from the format just choose run info and then create your file. So here you will get the already SRA run table if you are interested to process raw sequencing data. So let me just open this. So this kind of raw sequence, this is SRA run table. So this is SRA run table where we will get the information regarding each of these samples, and we can directly submit this SRA run table. So at the T-Bioinfo platform, so I'm not going to process this data, but I just want to show you how you can, once you have that SRA run table, so you must have in that text file. So let me just make it to the, I'm just taking the first two, three, this four samples. So you can take whole data. So I'm just interested to make only example SRA run table. So we can copy it in the Excel and we have to delete the last file. And now I can save this file. So I can, at the desktop, I'm saving this file as example SRA run table. So I will show you how we can, if you have not already processed data, then you can perform any analysis using this SRA run table. So already with this study, we have this, already we have processed data. So authors have already provided their pre-processed data in the read cons. So you can download this data, and let me just show you exactly what this file contains. So I have just taken this file and just then removed all those samples which having zero value. So once we have read con data, what we can do, so you have this data, if you're able to download this file and just remove those genes having a zero value. So after removing the zero values, so next what we can do, once we have read con data, so here, so I'm going to again the area of analysis. From here we can go directly to the unsupervised machine learning or the utility section. So once we have that gene expression table, so from that I'm not considering all the, let me just show you meanwhile. So let me just show you exactly what this table contains. So I'm just pasting it in the Excel here. Sorry. So if you look at this data, here you will find a different drug treatment. Like here in the first, we have this particular sample, then they have also drug treated. Mock samples, so then they have a particular drug treatment. So I, from this I have already selected a particular, only because there are multiple drugs, so I am interested only one data set that contain actually drugs treated. So this series 16 is actually, so let me just highlight here. So there are the different other drugs. So I have taken only this sample data, this part where we have COVID-19 lung samples, the simple normal samples. Then we have this, sorry, this is the 15. Okay, from series 16. So here we have 3 mock samples, COVID-19, and that are actually treated with placebo. And then we have 3 samples that are untreated. Then we have 3 samples which are ruxolitinib treated. So I have taken this data and make a new data set with and removed only all those genes which having 0 value across all the samples. So I have considered only important genes. So finally, I am able to produce a gene expression table having these 3 types of sample. One is mock treated, that is a kind of placebo, and then we have untreated samples, and then we have 3 samples that is treated with the ruxolitinib. So then now I have make a data set based on that, and then we can process this data depending upon the analysis that we are interested. Then we can upload this data and perform PCA. Whenever we have a gene expression data, the first thing that we should do, that we should visualize our data using PCA, so that we can understand what are different patterns within our data. So quantile normalization, as this is already PC normal all read count data, so I'm not performing quantile normalization. So we can directly perform PCA. So here we can select the threshold. So let's take it by default, and we are not changing any other option. So it's asking whether you want to perform log scale transformation. A read count data, we can skip this and just generate PCA component. So the PCA for the SARS-CoV. So once you have read count data, what other analysis that we can perform? Anybody can tell me? If you have read count data, what kind of statistical analysis that we can perform? So can anybody tell me, if you have a read count data, what kind of statistical analysis that we can perform? Whether we can apply t-test or not. So you can type yes or no, whether if you have a read count data, whether you can apply t-test or not. So for the read count data, we cannot apply t-test because t-test can be applied when we have our data have normal distribution. So what we can do, that we can perform differential gene expression using DSEC. So before performing DSEC, let me show you exactly what we have obtained by running this pipeline. So here we have obtained these results, PCA results. So now you can visualize your PCA plot using a scat. So based on this, in this data, so we have taken only there are three types of samples, as I have already told you. We have SARS-CoV mock samples, we have SARS-CoV-2 sample which are untreated, and SARS-CoV which is RUX treated. Then we have obtained these two PCA components. Let's see based on the PCA components how we can visualize our data. If you go to the insert and then go to the scatter plot, and here, so if you look at this data, so it looks like there are the three, if you look accept this one, there are the three different groups or the clusters are there. So now let's look at exactly what these samples are. So we can, so you just need to go to the select data, go to add, and here the first we have SARS-CoV-2 mock samples. Then we can select exactly x-axis value. If the SARS-CoV, this mock sample, these first three value, PC1 component as x-axis and PC2 component as the y-axis. So add another. Then we have untreated SARS-CoV-2 untreated. We can name it as untreated. Then we have x-axis, that is for the PC1 component value. Here, from here we can choose that. And for the y-axis, sorry, again, yeah. So again, we are left with the last. So let me, okay, select data. So here we have RUX treated. So the RUX SARS-CoV-2 RUX treated. RUX treated, ruxolitinib, series X. Here we have, this is X, PC1 as x-axis and PC2 as y-axis. So now if you look at this, here we have, based on the PCA components, we can at least clearly visualize that SARS smoke samples, here the red one. So one is look like outlier. Then we have this green one is untreated and the blue one is RUX treated. At least the two RUX treated samples are actually, we can visualize the difference between that. So is it clear? This is the first analysis. If we have readcon data, at least we can understand there are the three different patterns within our data. So we can understand that we can perform our on the because here we have used almost all the genes except the having a gene with a zero value. So we would have nearly 15,000 genes. From based on the 15,000 genes, we have just generated PCA components, and then we have tried to visualize the distribution of the samples based on the PCA components using all the genes. So the next step, we can also identify important significant genes. How we can do that? So here you will get this option. If you have read count data, you can perform AGR or the DSEC to identify only significant differentially expressed genes. So here if you just click on here, you will get this information as well. So you need to provide, please note, it is recommended to upload expression in the raw read count values. So DSEC or the AGR only can be applied when we have read count values. So if you have already normalized like FPKM values, so in that case, you can apply t-test. But if you don't have already non-normalized value like read counts, you can perform DSEC and the HR. So then upload your data. So here we are considering only two groups, treated and the untreated, because differential gene expression analysis can be applied only on a two conditions. So here I have deleted already mock sample. So I have just considering treated and the untreated groups. Then we have to define which are sample belong to which category. So instead of the 9 sample, now I have only 6 sample. 3 belongs to the untreated and 3 belongs to the treated group. So here we can, this ruxolitinib group, we can just drag this sample here and untreated sample as group A group. So then we can submit and then we can go over there. So then we can perform DSEC. Here we can filter out all the values like already prior the normalization. We can filter out how much cutoff that we are required. I'm taking this as default, but here I'm selecting a p value less than 0.05. So I'm considering a p value 0.05. So then you can also change this log fold change threshold for the volcano plot, and then you can also select if you want to perform any to check go enrichment or kind of things, so you can make that cutoff as well and save this and run this pipeline. So it will take a bit time, like 3-4 minutes. So this is how we can directly download our data from the public repository, and then we can perform any analysis using T-Bioinfo platform to drive a significance meaning. Beside, you can also run a, meanwhile, let me just show you. And we can also perform any other unsupervised machine learning. As we had during the session, we have also gone through the unsupervised machine learning, that is hierarchical clustering. So let's see how we can perform hierarchical clustering on this data. So I have just used the same data that we have run during the used for the PCA, that we have 9 samples with the read count value. So let's go over this hierarchical clustering. So as I know, there is 3 types of samples. So I'm making a cluster maximum 3. And as we have to do the transpose, as we want to the cluster the sample. So HClust for SARS-CoV-2. have to run the pipeline. I just forget to click on this. So then already my DSEC pipeline completed. Now let's see what the output that we have obtained after running the DSEC. So if you look at here, we have already get the first this all DSEC. So let me just show you exactly what we have obtained. So then here we have volcano plot, we have MA plots, and we have this log fold chains plot. So let me just show you exactly what output that we have obtained after running that particular pipeline, DSEC pipeline. So if you look at here, so now based on here we can add a filter. So in the DSEC, if you remember the last time, we have usually obtained p-value, p-adjusted value, log2 fold chains. So we have obtained this kind of data. Now you can filter out only the significant genes. So that is p-adjusted value less than 0.05. You can filter out and understand what are these genes and what are important genes that can actually discriminate treated and the untreated group. So if you look at based on the p-adjusted value, so already we know that p value should be less than 0.05. So if you look at here and you consider only those genes having a p value less than 0.05, so here we have nearly 474 genes that having p-value less than 0.05. So you can find out what are important signature within this data that can actually discriminate or distinguish this these two groups, treated and the untreated. So then if you look at the other outputs that we have obtained, so let's look at this. This is the volcano plot. So here we can get how many genes are there that is actually uh, significantly differentially expressed in these two conditions. So this was the volcano plot, and then we have this MA plot that represent how many genes are actually having particular log fold genes and how much mean normalized value for all those genes. So this is what we can run and exactly try to can find out important genes. Then we had run this other pipeline as well, hierarchical clustering. I think that is also completed. So let's see exactly what we can done. So if you look at hierarchical clustering, this I'm taking this dendrogram directly. So if you look at here, we have these MOG2 samples that are clustered together. Then we have these two RUX treated groups. So these are clustered together. One RUX treated group is actually clustered here, and one MOG sample is also clustered differently. So then we have these SARS-CoV-2 samples, and these are actually which is untreated sample. So based on from the public repository data, we can perform various analysis and can find out important genes. Now you can also perform TCA and hierarchical clustering with only significant genes. So those kind of analysis we can done, and this is the major public repository GEO or the SRE from which we can obtain RNA-seq data. And if you have any questions till now. So do you have any questions? So during you can submit your project form as well if you're interested to perform to execute any project and to interested to design your research project. So you can go for that. And if you're interested in RNA, sorry, the faster data, that is sequencing data, so you can take those data from the NCBI viruses. So you can submit your project form, and then if you're interested or you have any questions, still you can post in the forum. So is there any questions? So is there any question? So if you have no questions, okay, okay, Utkarsha. So if you have no questions, so we can proceed for to close this session. And if you left with any query, you can always email us, and you can post your query on the forum as well. So thank you everyone for joining today's session.